Thank you, Donato, and thank you all for being here. I'm really excited to have the opportunity to talk to you about my work and the work of many of my colleagues in mineral evolution and the coevolution of the geosphere and biosphere. And much of the work you're going to be seeing here today is coming out of the Keck-sponsored Deep Time Data Infrastructure Project, which has a great group of PIs, including Bob Hazen and Peter Fox, and also Paul Falkowski, who's here today and is going to be giving us the keynote address tomorrow. So one of the main aims of this project was to better understand the complex co-evolving systems from a data-driven perspective. All too often, biology and geology are treated very separately. But as everyone here in this room well knows, that's the wrong way to go about it, because life emerged and evolved in the presence of rocks and minerals and fluids. And likewise, life had a dramatic effect on the chemical landscape and therefore the geology and geochemistry of our planet. So if you're more interested in this project, you can certainly check out our website. But the main goal of this project was to have a more holistic perspective of the evolution of our planet by integrating the large and growing geologic and biologic data resources in order to make discoveries that one field alone cannot. So this project has been particularly exciting for me as a mineralogist, not a biologist, as you will learn by my talk, but it's been particularly exciting for me as a mineralogist because largely for a long time, mineralogy has been a very descriptive field. We essentially go into the field or we synthesize minerals and we measure them and we describe them. But we've largely for a while been somewhat removed from geology and thinking about evolving systems and certainly far removed from life. So it's been really exciting to me to see us go into a more predictive phase where we can do things like predict the number of missing mineral species using these mineral ecology studies. We're also working on projects where we can pinpoint the specific location on Earth's surface of minerals, and likewise of knowing what minerals we expect to find at any specific location. Something that's going to be perhaps of more interest to everyone in this room is that we're also exploring the idea of Earth's mineral diversity and distribution as a planetary scale biosignature. And I'm going to talk about that some more. So we're not only making advances in mineralogy, although I'm skewed towards that, but we're also making advances in things like tectonics and the supercontinent cycles, in paleobiology, and in petrology. So Eric asked us to think about some big questions that we can address regarding this bridge between Earth and life. So specifically the things I thought about were, what do Earth's earliest environments look like? That's something that we've been talking about a lot, right? And how do we actually tease out that information? Next, do network topologies of minerals, fossils, proteins, et cetera, embed timelines? Now, many of you geologists, chemists, you're a little bit more up on, biologists and chemists, you're a little bit more up on using these kind of advanced analytical techniques. Geology is slowly trying to catch up with you. So we're learning a lot now about embedded intrinsic timelines and other features like chemistry and extinction events that we can get out of these analytical techniques. Next, as I mentioned before, is the mineral distribution that we're seeing on Earth's surface, a planetary biosignature, and can we use that in planetary evaluation and exploration? And lastly, and I really hope that this is true, can geologic data reveal information about the emergence and evolution of the earliest organisms? So in my talk, I'm going to stick to three topics. The first is data resources. I'm just going to go over that really quickly. Mineral evolution, and mineral network embedded features that we've been observing. So first, data resources, just to show you a few to give you some context of what we're working with. The first is the RUF project. So this is one of the largest mineral databases and libraries in the world. It's founded by my PhD advisor, Bob Downs at the University of Arizona. And it provides a set of very high quality spectral, chemical, and X-ray diffraction data, in addition to some pictures and some references. Currently, we have around 3,500 distinct mineral species that we have characterized. Just for reference, there are around 5,300. So we've done over half, and that number is continuing to grow. Right now, we have over 10,000 mineral species in the database and library. And one of the great benefits of this is when you're looking at this chemical information and you're comparing it to the X-ray diffraction information, you know that those two pieces of information were gathered on the same sample. As you know, minerals show a lot of variation from sample to sample, and certainly from location to location. So it was important to us to make sure that we were collecting all of this information on the same sample, so it's all consistent. As a part of the RUF project, we have the list of officially approved minerals from the International Mineralogical Association. And on this website, you can search for specific phases if you'd like. Like I said, there are around 5,300 of those today. Of course, that number is continuing to grow as we find more and more minerals. You can also use the searchable chemical feature. So if you're interested in finding minerals that specifically have a certain composition or don't have a specific element, you can also search on these tags here, and there's a lot of information you can get to from there if you're interested. What I specifically want to tell you about today, though, is the Mineral Evolution Database. So that's this button right here. And this database was started a number of years ago by Bob Hazen. And what you see in this database, and this is just a tiny glimpse of it, it represents countless hours of literature review, most of which has been done by Josh Golden at the University of Arizona and a number of undergraduates that he's trained in this process. And what we have here is locality information, so geologic localities and the minerals that occur there. And often, and the goal is to also have ages associated with that. So currently, we have almost 1 million mineral locality pairs, and we have over 120,000 mineral locality age data. So that's quite a lot of data for minerals. The next website I like to mention is mindat.org. This is a crowdsourced website that gives geologic locations, some interesting information about them, but what we're really interested in is the fact that we can extract what minerals are present at that location. Right now, they have around 300,000 localities and about 1 million mineral locality pairs. So we also have a lot of data coming from here. Now, I mentioned that this was crowdsourced, and maybe this gives some of you pause. It did me. I was concerned about the accuracy of this data. But as anyone who works with really large data sets is familiar, errors tend to average out. Now, of course, while we feel that we're working with big data for mineralogy, we recognize that these numbers are not very big and compared to the statistics that are generally being done. So it was really important for us to test that the errors truly were being averaged out. So I mentioned the missing mineral predictions we can do earlier. So what we did was we ran that model on a mineral data set that was strictly taken from peer-reviewed literature. So absolutely confirmed mineral occurrences. And then we compared it to what we found with MINDAT. And it turns out that they produced the same result, even on relatively small subsets of the data. So we're pretty confident in using MINDAT, although we're still certainly careful in anything that we do. The next one I'd like to mention quickly is EarthChem, ran by Kirsten Lenner at Lamont. And this is a great geochemical database where you can get bulk chemistry, trace elements, and ages on a plethora of rocks. It's been huge for our tectonic studies. PaleoBioDB, Drew Masenti and Mike Meyer have really utilized this fossil network and have also contributed to it. And with that, I will get out of the resources and I'll talk a bit about mineral evolution. I think a number of the people in this room are probably familiar with this idea, and maybe you've heard Bob talk about it, but I wanted to mention it today because I think it gives us a good framework for what we're thinking about in this symposium. So first, mineral evolution focuses on changes in Earth's mineralogy through deep time. Now, I know that I'll probably get the question about the term evolution, so I just want to go ahead and say straight away, I'm not referring to Darwinian evolution, I'm using a much simpler definition of the word, which is simply a system changing through time. So that's what I mean. And that change through time happens as a result of new minerals forming through a combination of chemical, physical, and biological processes that are different at each stage of planetary evolution. So first, in the earliest minerals, once the supernova cooled enough to start forming some very nano and microscale grains of minerals, we had about 12 distinct mineral species. So things like diamond, graphite, moissanite, just very few. Once things started to cool a little bit further and we're forming planetesimals, we can get up to around 60 minerals. And then once our planet is finally cooled enough to form a very primitive crust, we get up to around 500 minerals. Through further igneous evolution and differentiation, we can get up to 1,000. And then with the onset of plate tectonics, that helps us achieve 1,500 distinct mineral species. So at this point, we've gone up through about 2.5 billion years of Earth history. But I said earlier that there are 5,300 mineral species today. So what happened in between that 2.5 billion years and now to give us this more than a threefold increase in mineral diversity? And the answer to that is life happened. Oxygenic photosynthesis, specifically. So as you're all familiar with, here is a graph of the oxygen fugacity through time. And we can see at 2.5, the great oxidation event, we see this dramatic increase in atmospheric oxygen. Now, this completely changed the chemical landscape of the surface of the Earth, and it had a profound effect on the mineralogy. So just to give an example, here we're looking at copper minerals, and the oxygen fugacity of our environment, of our surface, was right at this line prior to the rise of atmospheric oxygen. And what you can see here is it would have been very difficult to have the widespread formation of copper 1+ or copper 2+ mineral species. In fact, more than 400 of the 700 known copper mineral species would not have formed in those pre-GOE conditions. It also means that things like copper 2+ would not have been available for life to utilize. So if we look at the mineralogical record, we see that that trend is confirmed. Here we have a max age on the axis, on the x-axis, decreasing to the right. And then on the y-axis, we have mineral occurrences. So we can think of this as sort of like abundance. We don't have mineral abundances because this is actually very hard to estimate and determine, although we are working on that. Basically, mining companies know how much of their minerals they have, but aside from things that are not economically important, it's really hard to estimate. No one's really studying that. So for now, we're going to use the number of occurrences on Earth's surface. Also, in these blue bars represent copper 1+ minerals, and the green bars represent copper 2+ minerals. And what I'd like you to focus on, there are a number of things we can see here, but what I'd like you to focus on is the relationship and the ratio between the green bars to the blue bars. In other words, the oxidized copper species to the reduced copper species. So let's just zoom in a little bit. And what I'd like to point out that as we see a rise in atmospheric oxygen, we see a steady increase in that ratio of oxidized copper phases to reduced copper phases, which is exactly what we should see, right? There are a number of other things you might notice here, questions you might have. For instance, why are these bars so much lower? And a lot of that has to do with erosional bias. How to quantify erosional bias is a much more difficult question to answer, and that's something that my colleague Qiao Liu and Simone Runyon are working on, trying to figure out how much of what we're seeing here is erosional bias. And also you'll notice that we have peaks associated with the supercontinent assemblies. And this is a really fascinating thing to think about. I'm not going to go into it today, but my colleague Qiao Liu just had a paper come out in Nature Communications looking at this, thinking about pulses of mineralization associated with these supercontinent assemblies, specifically thinking about Rodinia, because it turns out that the Rodinian supercontinent behaves a little bit differently. So if you're interested in that, please check out his paper. And if we look at the other first row transition elements, we see a similar trend. So we're looking at things like iron and manganese and cobalt, and we're seeing that these exhibit really similar trends to what we saw with copper. We went as far as to generate these diagrams for the whole periodic table. I don't expect you to be able to see this, and we found that by and large these trends are true of most elements. There are some notable differences, as you would expect. And if anyone's interested in actually exploring this, each one of these images is a high-resolution PDF, so I can send this to you, just let me know. Now, obviously I care about minerals because I'm a mineralogist, but why should you care about minerals? Aside from them just being generally interesting, they also provide ancient samples. So we don't have direct samples of the DNA from earliest organisms, right? We don't have examples of, or we don't have direct samples of the aqueous chemistry from those time periods. But what we do have are rocks and minerals that formed during those time periods. And in their formation, they tell us a lot about the conditions that were ongoing during that time, the chemical, temperature, pressure conditions. So we can tease out a lot of information about what was happening during the time periods of interest simply by looking at rocks and minerals. So we think we'll also be able to characterize bioavailability of these elements through time. For instance, when copper 2+ became available. And we hope that we can also relate this to the timing, relate the timing of elemental availability to the emergence of biological functions. I'd like to mention one paper here by Eli Moore, and you'll notice some names of people in the crowd here, Donato and Paul, a paper that is thinking along these lines, and I think that some of this work could contribute to their work in the future. And they're looking at the metal availability of microbial metabolisms, and essentially they reconstructed the history of this metal utilization by identifying links between redox state, metal availability, and metabolic pathways. So this is something that we could hopefully help with in the future. One other study I'd like to point out is by Janne Bromberg at Rutgers, thinking about protein evolution. So here, she's constructed a network of the structural similarity of metal binding sites. And what is interesting and striking here is, despite the fact that she didn't actually include the metal in the layout, when you overlay it with the color, you see this very distinct line from iron to manganese to copper. And she thinks that we're actually looking at a timeline here. But of course, we're looking at a relative timeline, right? We can say, okay, well, iron came before manganese, came before copper. But I hope in better constraining the availability of these elements through geologic time, that perhaps we could help her put some absolute ages on this rather than just relative. So we're working on that with her and hope that we'll be able to do that. Now, the last section I'd like to talk about is some of our mineral networks and the embedded features that we've observed therein. First, we have a network here of copper minerals. So each of these nodes represents a copper mineral species. They're colored according to their chemistry, so the presence or absence of sulfur and oxygen. So the red phases are sulfides, the blue are carbonates and oxides, and the sulfates are in yellow. The nodes are sized according to the number of their localities, so again, we can think of this kind of like abundance. And the links are scaled inversely proportional to the frequency of occurrence, so if they occur together often, they are closer together. If they occur together less often, they are farther apart. And so the thing that immediately pops out here is their high degree of partitioning on chemistry. And in fact, we can actually draw an oxygen fugacity line directly onto this network and onto the data behind it, and likewise a sulfur fugacity. And we think there are a lot more bits of information that we can tease out of these networks. Next, here's a similar copper network, and here we're looking at a structural complexity. This is work that has been pioneered by Sergei Krivovichev at the Kola Scientific Institute for the Russian Academy of Sciences. And here he has come up with a method for quantifying the complexity of mineral crystal structures and reporting that in bits of information. And this is particularly interesting because he hypothesizes that minerals began as very simple structures and over time became more and more complex. So this is something we're working with him to try and think about. Is this true? And so if we look here at this network, you can see the very simple and the simple structures tend to cluster more or less in this area, while the intermediate tend to cluster here. So we can say more or less, we're seeing there's a trend. But what's interesting is that that logic seems to fall apart when we're talking about these more complex and the very complex phases, things like clay minerals and stuff. They tend to be just kind of interspersed throughout the network. So I'm not sure what to make of that. So it holds to a certain point, and then we're losing it. So this is an ongoing study. The second, or the last network that I would like to show you is that of carbon minerals. And so here it's a slightly different network. It's bipartite, so we have two different types of nodes. Again, the colored nodes are carbon minerals. They are colored and sized, in this case, and according to their number of localities. And the black nodes represent the carbon mineral localities on Earth's surface. So, for instance, this is the Kola Peninsula in Russia, where we have a lot of carbon mineral species. And these nodes are sized according to their mineral diversity. So if the black node's larger, it has more species. And I bring this up to make 2 points. One is that we can explore those planetary scale biosignatures that I was talking about earlier. And the second is to show you an embedded timeline. So first, let's look at the biosignatures. So here we're seeing a graphical representation of the distribution and diversity of carbon minerals. If I were to show you one for all minerals on Earth, it would look quite similar to this. And this distribution conforms to a large number of rare events, or LNRE distribution. And this distribution is characterized by the fact that most minerals, like kozawite, which is a lanthanum carbonate actually found here in Japan, are rare. And only a very few of them are common, things like calcite and aragonite. And so in this distribution, if we show it in a bar graph, follows this trend right here. And with this model, we're actually able to create an accumulation curve and predict the number of missing carbon mineral species, or whatever set of minerals you're interested in. In this case, when we developed this data in January 2015, there were 403 known carbon species, and we predicted that there were 145 missing carbon minerals. And if I can digress a little bit, this actually spurred a citizen science project called the Carbon Mineral Challenge, where mineral collectors and scientists alike were encouraged to actually go and find these 145 missing carbon minerals. That project started in 2016, and as of last month, they'd found 12 new carbon mineral species. So that's nearly 10% of what was predicted, and a number of these species were actually directly predicted in the paper. So it's been exciting to see this actually happening and making predictions, and they're happening. So now if I can come back to this biosignature idea. Again, we're seeing this graphical representation of the distribution of minerals. And as I said before, Earth has around 5,300 mineral species. Now, if we look at other planetary bodies that we have a feel for their mineral diversity, we don't find, and granted that data is limited, we don't find that they have quite this many. In fact, they usually have on the order of hundreds rather than on the order of thousands. And they certainly don't have these extremely rare species. It's more these things that are just quite common. This is certainly true of Mars, where we can look at meteorite data and we can look at rover mission data. And we found that, it's certainly less than 500 minerals, and we don't have that large amount of rare mineral species. This is also true of the moon, where we have Apollo mission samples, where we see around fewer than 350 mineral species. So what we know that this distribution on Earth happened as a result of the rise of atmospheric oxygen due to life. Now, and we know that when we look at planets or bodies that presumably didn't have life, or didn't have advanced life, that they don't look like this. So the idea is that perhaps this signature is a planetary scale biosignature. We don't know if that's true, but we tend to think that that is the case. So that's something that we always have in the back of our mind when we're doing these studies, and hope to pursue that idea further. So the last thing I would like to mention on this is that here we have, it's the same copper, it's the same carbon bipartite network, but I've colored the nodes a little bit differently. So you'll see here they're actually colored by age now. So with red being the oldest, moving into blue being the youngest. And you expect when you're looking at biologically evolving systems that there is certainly going to be a timeline. However, I didn't necessarily expect when we were looking at mineralogical systems that we were going to see a timeline. However, it's pretty clear. You can see we start here in the center of this locality U with the oldest red minerals moving up into orange and yellow and green, finally out into blue on the outskirts. So we have this nice timeline that essentially just fell out of this network. And we weren't really expecting that. So with that said, Eric asked us to think about where we thought Earth and life science was going. And basically what I came up with is the fact that Earth's evolution has been an intertwined succession of increasingly complex physical, chemical, and biological events. So I hope that with this talk I've shown you that I think the key to understanding the complex history of our planet and other planetary bodies is the continued development and integration of cross-disciplinary data and expertise, which is exactly what we've been doing with the KEK project. And in fact, we believe that so much that we've actually gone on to organize another workshop, if I may plug it now, called the 4D workshop, which is the deep time data-driven discovery of the evolution of the Earth. And in this workshop, we're bringing together a diverse group of geologists and biologists and data scientists from around the world to come in and to think about the emerging methods of data analysis and visualization and how that can help us understand the past, present, and future of this evolving system. So with that, I would like to thank ELSI for inviting me to come and share my work with you today, and to our sponsors, and certainly to my collaborators, whose a lot of work I presented today. So thank you.
Questions? Yes, I'll start with John and I'll get back.
Thanks for that nice talk. When we see a plot of anything, abundance with age, particularly when we see things being more abundant in more recent times than in previous times, there's two ways to interpret that data. One is that it represents more recent production of those things. On the other hand, you could say, well, maybe this is a preservation artifact, that this represents that things that are older just didn't survive, they decomposed. And what I'd like to know is, how do you separate these things? Because, when you show the periodic table, all those curves are increasing, and I guess my null hypothesis would be to interpret those purely as preservation artifacts, not as production.
Right.
So how do you go about doing that?
Well, I would love to know how to do that as well. So we're certainly looking for collaborators and people who have ideas on that. So if you're interested in helping us explore that, we would love to. As I mentioned, my colleague Chao Liu and Simone Runyon at Carnegie are actively working on trying to quantify that. So in addition to having this erosional bias, this preservation bias through time, we're losing things, old things. We also have a lot of ephemeral minerals, things that are very soluble, that we see, we see this huge increase in them. Likely, they certainly were forming back then, but you will never see them. So it's not even a matter of, we've lost all of that rock. It's a matter of they're soluble. So they're just going to disappear. So there are a lot of things that have to be considered. Certainly that general upward trend is largely the result of erosional bias. And so it's important for us to think about how, were these more oxidized phases more affected than the reduced phases? That's certainly important when thinking about this trend. So yeah, it's a great question, and I would love to have a better answer than that, but we're working on it.
Okay. Okay. Next, my question is related to John's questions. For example, early Archean rocks, it occupies early Archean rocks like Greenland, Eastern Labradors. It's 3.8 to about, say, 4 billion, very close. Occupy only 0.01% among the total landmass. So it's really small. So that therefore, next question is how to reconstruct the mineral distribution and the species. So the one idea is that all of those rocks is a kind of accretionary complex. So the accretion complex means the fragments of oceanic crust and also trench turbidites. So that and therefore this is what we call Pacific type organ. It's quite different, much bigger geological units. Like, continents are formed by subduction, just granitic rocks. And also passive margin, like off North America, is off New York. So completely different set of rocks. It means completely different kind of minerals, right? So that therefore my recommendation is you should first, to make a database, you should first to put intermediate, for example, rock type. What kind of orogenic belts and formed by what kind of process?
Right.
So
That's
that,
what you mean, yeah.
this is one method how to extend. And we are very much in the Hadean rocks. Hadean rocks is completely absent on the Earth now, but it must be present. If it is komatiites, formed at mid-oceanic ridge or whatever, it's because due to high mantle potential temperature. So therefore, basalts at hydrosurface doesn't work at all, because completely different set of rocks in the Hadean. So how to estimate the minerals and also the mineral abundance and others, and also water rock interaction is completely different, field, right? So my next question or advice is the localized environment is completely different. For example, oxygen in ocean and atmosphere. Even today, Mount Fuji volcano is basalt, flows to involve the trees, and then we will have native iron, even under extremely oxygenic atmospheric environments. So that therefore you should carefully classify the mode of occurrence and the process, if available data.
Right.
So that therefore these two, global or local, and also what kind of orogenic belts and through geologic time, and the goal is Hadean.
Right.
So how to estimate?
Yeah.
So this is my
advice. Thank you.
Yeah.
I really appreciate that. And you're really touching on the complexity of what we're up against here when we're dealing with not having often, direct access to these samples. And I really appreciate your comments. That's very insightful. Thank you.
So my question is, so first it's a bit of a silly question. We've barely scratched the surface, literally, of Mars and the Moon. So I found it a bit striking, the numbers that you say, for such low numbers for both bodies. So it may take a while, because we're still finding 12 minerals until last month on the Earth. So is it possible that we just haven't sampled enough up there? And relatedly, if those are the numbers, then may the such mineral poor state of Mars basically prove that it never had life?
Well, so I actually work on the Mars Science Laboratory Curiosity rover, and one of the questions that we often get is, so was there life on Mars? Is there life? Or, do you know, and you're just not telling us, these sorts of things. And what I tend to think is this lack of mineral diversity that we're seeing with the CheMin X-ray diffraction instrument could indicate that we didn't have advanced life on Mars. Now, could we have had some more basic life? Perhaps. I don't know enough to say whether or not that is true. But I tend to think that... Right. So, yeah. So I tend to say when people ask me if there was life on Mars, I tend to say no, which is kind of a bit of a bummer, and perhaps some of my Mars colleagues might not like for me to say that, so maybe don't air that part. But I tend to think that there wasn't. But I absolutely could be wrong, and I'm totally open to other hypotheses. So to get at what you initially said, yes, perhaps we just haven't found it yet. But for instance, on Mars, it's very likely that there's not much evidence for granite crust formation or certainly plate tectonics and things like that. So the likelihood of generating quite as many mineral species is automatically diminished from that standpoint. But still, if we're thinking about life dramatically making these chemical niches, I would expect to see a much higher number than what we have. We only have a few rover missions that have gone, and we only have a few Martian meteorites. So it's a big planet. And likewise, the moon is also really large. So I think we need way more exploration before we can say anything definitive about that.
Perhaps would be also interesting to know how much of the minerals are exposed to the surface and those are accessible to those techniques compared to what in reality is down.
Yep.
So we have Caleb and then I'll come back. We still have 5 minutes.
Well, you didn't throw it at me. So I think this is wonderful, and this may be a very difficult question to answer, but I'd be interested to know your thoughts on it. So one of the things that strikes me is that when we talk about biosignatures, whether it's Earth, Mars, wherever, we don't have a particularly good calibration as much as we don't know what a purely abiotic Earth would be mineralogically after 4 billion years.
Right.
Neither do we know what a purely abiotic Mars should look like. I don't know what the answer to that is. I wonder if you had any insight, whether there's some way in mining the data, looking at persistent ratios of particular minerals, whether there's some clue to what a purely abiotic planetary, terrestrial type planet would look like after billions of years.
Yeah, I mean, I think it depends on the pathways that you think the planet went in, as far as its chemical environment. But yeah, I mean, you know, many of these minerals that formed, many of these 5,000, this huge boom, could have formed abiotically. A number of them couldn't have. So the first order step is to actually just remove anything that could not have formed without the direct presence of life. We still have a huge number, so that doesn't really help answer that question. You can make some assumption, you can cut it off at plate tectonics and say, okay, well, but that doesn't really make sense, right? Because the chemistry of the planet is going to continue to evolve and you're going to create new chemical niches. So you're right, that's a very difficult question. And it's one that we think about a lot, but I don't have a good answer for. Thank you.
Hello, I found your talk absolutely fascinating.
Thank you.
One of the questions I had was, I found it interesting that there was a great rise in mineral oxidation at the onset of the Cambrian. And have you guys looked into any historical bioturbation data for mineral exposure in terms of mass extinction events? Because if there's burrowing creatures that go under the oceanic, crustal surface, then does that mean that there's more mineral exposure, creating more oxidation for minerals?
Yeah, sure. I mean, in general, you're going to get oxidized material within the first, few kilometers of the crust relatively quickly through abiotic processes. But I certainly think that biotic processes could influence that. I mean, they definitely change the mineralogical landscape of any environment. And in fact, I didn't show it today, but I have my colleagues, Drew Macenti, and Mike Meyer, that I put up there, have done a lot of work in thinking about paleobiology. So if you want to see me afterwards, I can show you a lot of the work that they've been doing. We've got some cool manuscripts that maybe they won't mind if I share with you.
So I would like to ask about the connection between your talk and Everett Schock's talk. So you showed this increasing mineral diversity. How does that relate to the way that life is solving Earth's problem in terms of the thermodynamics? So in other words, how does that show that, in a sense, the life is helping the planet approach some more equilibrium state than initially? Could you have something to say about that?
Yeah, so essentially life is creating these widely varying chemical niches, right, all over the place. They're making it very strange chemically, and that's actually what's creating these environments for these different minerals to form. That's what's creating this huge mineral diversity. So aside from that, I'm not sure that I can really directly relate it to Everett's talk, although he's raising his hand, so I think he might be able to do that. So I would love to hear what Everett has to say about that.
So I'll get to Everett, and then we have two last questions, and we need to move on, unfortunately.
I think there's a major difference in that a lot of what you see is driven by photosynthetic oxidation of the Earth's surface. And that is a different thermodynamic state than what I was talking about, where I was talking about what the planet provides chemically in terms of chemical energy sources. It's a different. There's a serious thermodynamic consequence to drawing your box to also include the sun, or at least solar photons, and as a result, then what we're familiar with at the surface of the Earth, especially walking around the continents, is a lot of disequilibrium generation by life.
Yeah.
But that's because of cheating and grabbing those photons and working uphill and producing dreadful conditions like a 20% oxygen atmosphere, where that's a horrible place to live.
Extreme conditions.
Yeah, extreme conditions like that. So there is a difference. But it's a different. There's major consequences once phototrophy begins.
Right.
I'm getting there, Paul. There's one question before you, and I'm coming back. The room is big.
Thanks, nice talk. I just had two, fundamental questions for how you analyze your data. And one of them has to do with the threshold to have a mineral. Was it just simple presence or absence in terms of any abundance of the mineral when you plot it on the networks? And also for drawing the connections between two minerals, I assume it's like co-location, but can you talk a little bit more in detail about how you made the cutoff for if two minerals were co-located or not?
Yes, absolutely. So to answer the first question, and both of those questions are something we think about a lot, and there's significant problems in handling our data. So firstly, ideally, we would have abundance information, right? I would know exactly how much pyrite by volume, by weight, however you want to measure it, is on Earth's surface, but we don't know that information. We have somewhat of an idea for economically significant things, but we don't for things that mining companies don't care about. So for now, we are basically, if MinDat or if a piece of peer-reviewed literature says that this is a geologic locality, we're calling that one occurrence. So it could be that there was one crystal there, but it could be that there was a ton there. So that is a big flaw in using the number of localities instead of abundance, and that's something that we're trying to get past, but it's a really big hurdle. And co-occurrence. So co-occurrence is if they were cited by the literature as having occurred in essentially the same geologic unit, geologic setting, which there is some variation there, and that's something we have to think about very carefully with each sample, which is why that mineral evolution database, when I said it represented countless hours, I really mean countless hours. Josh Golden has spent, I think it's about 6 years now, maybe a little bit more, working on that database. And for him, that means painstakingly going through every piece of literature that he could possibly find, and really understanding the geology behind the statements that are being made in that literature. So it's not a perfect system, but
So Shaunna, it's not necessarily a question for you, but maybe you and Everett and others in the room. Dante Keneal and others have painstakingly tried to understand whether there's been a net oxidation of the crust and mantle through time. And using chromium, vanadium, others, the valence states of these hasn't changed. So there clearly is superabundant amount of reductant within the mantle.
Yeah.
It begs the question then, do you really care whether you have to go half a kilometer into Mars or just scratch the surface? Because you're looking at the surface manifestation of mineral evolution, really.
Right.
So it's the thin skin, right?
Yep.
So that makes life a lot easier for you if you can justify that with, and especially because mantle convection is virtually nil on Mars today. I mean, I don't know if you have thoughts about this, but more fundamentally, I guess more profoundly, is why hasn't the mantle changed, or at least the upper portion of the mantle that we can sample changed in terms of redox state through time?
Yeah. So your fundamental question, I think, maybe Everett wants to think about that. I don't want to try to touch that. But in thinking about Mars, yes, that's exactly my line of thinking, is likely when we're just looking at the surface, where we can do pretty much a direct comparison to what I'm showing here for on Earth. I mean, on Earth, obviously, we know a lot more about the mantle minerals, but as you pointed out, we find that extreme diversity is actually located in our crust. So yes, absolutely. Now, why the mantle hasn't dramatically changed as a result of this is something that a lot of people are thinking about,
Sure.
right? And yeah, I don't have a good answer for that. If someone does, I would love to hear it. Everett has something to say.
Life has had a profound impact on the atmosphere and a negligible impact on the oxidation state of the mantle. Mantle's big. Life's wimpy. Sorry about that.
That's a
And
solid answer.
let's not
Get over it.
yeah, get over it. Let's be careful to not equate life and photosynthesis somehow in our thinking here.
Yeah.
Yeah, but it's not very effective.
So if you have all this convection going on in the mantle over geologic time, it still hasn't changed the redox state of the mantle. It's just, that's because there's so much reductant in the material. Maybe it covered by.
The mass of the mantle is pretty profound.
I think we have another suggestion here, and then we need to move on. No? No. Well, let's thank the Shaunna and Everett again.